Okay, so this is going to be the first one in the medical microbiology playlist. In this first video, we're going to mainly be talking about the genus of gram-positive bacteria Staphylococcus. But first, we're going to go over a quick recap of gram staining and what it actually means. So a gram-positive bacteria will stain purple from the gram staining process because crystal violet is retained within the thick peptidoglycan cell wall. So if we look at this image on the top right here, we can see the kind of outer structure of the gram-negative and gram-positive bacteria. We can see the gram-positive bacteria has a lot of peptidoglycan in its cell wall. When you stain that with crystal violet, the crystal violet forms complexes within that peptidoglycan cell wall, staining it purple. So when we look at the bacteria at the end of the gram staining process, they're going to appear purple, because even after we apply safranin to it, the purple colour kind of overrides the pink kind of reddish colour of the safranin. So the gram-positive bacteria are going to stain purple, and the gram-negative bacteria are going to stain the pinky red colour from the safranin, because you can see here it has a thin peptidoglycan cell wall compared to the gram-positive bacteria, but more importantly, it has an outer membrane. It doesn't say it here, but that's an outer lipopolysaccharide membrane, and it's basically the capsule of these gram-negative bacteria. Now, it allows gram-negative bacteria to be more resistant to bile salts and other things in your immune system. But when we're gram staining it, when we add the crystal violet, it's going to form complexes in this outer lipopolysaccharide membrane, and then when we wash the bacteria with alcohol, which degrades this outer membrane, it's going to get rid of the crystal violet, and then when we stain it with safranin afterwards, you're only going to have the safranin in the peptidoglycan, which is going to give it that kind of pinky red colour. Now, I didn't go over the full gram staining process, I just wanted you to kind of know the difference in structure between the gram-negative and gram-positive bacteria. If you're confused about the gram staining process and you don't really remember it from high school or A-level, there's plenty of videos online of it, so I won't go into much detail on it. This is more of a module focusing on bacteria species and other kind of microbes. So, getting into Staphylococcus, Staphylococci are gram-positive bacteria found on the skin as well as in the human microbiome as part of our normal flora. So we, like in our stomach and intestines or on our skin, we pretty much always have the Staphylococci bacteria on us, but most of the time they don't infect us, because most of the time they don't have the facility to infect us. They only actually cause an infection if our kind of immune system barriers become broken, like if you damage your skin, or if these normal microbiota are kind of moved around from where they usually live in your body to another area where they shouldn't be. It can cause what we call an opportunistic infection, which we'll get more into later. So, staphylococci are typically transmitted via direct contact from fomites, which are just objects carrying pathogens, or other medical instruments in a hospital setting. If the skin barrier is broken or the microbiome is disrupted, for example, the microbiome can be disrupted by certain antibiotics, the Staphylococcus can cause an opportunistic infection, which can either be minor or it can be fatal, but it's very rare to be fatal. That's mainly with a thing we call necrotic fasciitis, which we'll probably go into in the next video because we're running out of time. So staphylococci are facultative anaerobes, meaning they can survive anaerobic conditions, but they tend to prefer aerobic conditions. And they appear as spherical cells clustered like grapes because they divide successively in different planes, which is different to the genus Streptococcus that we're going to go into in the future, which is another type of gram-positive bacteria, where while Staphylococcus form these kind of grape-like structures that you can see in this image here, kind of like random clustered like berries, Streptococcus form kind of horizontal lines because they can only divide on a single plane. And in case you're interested, it's a fact that staphyle is Greek for a bunch of grapes and coccus is Greek for berry. So if you kind of want to remember Staphylococcus, kind of bunch of grapes or berry, because that's kind of how they're arranged. So if you're ever trying to remember the kind of structure of Staphylococcus under a microscope, just think of a bunch of grapes, which is convenient. They're also purple and you can get purple grapes. Staphylococcus tend to be non-motile and resistant to various conditions by being resistant to salts, desiccation or drying out, resistant to radiation and temperatures up to 60 degrees Celsius. And this is because of their thick peptidoglycan membrane that we talked about up here. Staphylococcus also produce catalase enzymes which break down hydrogen peroxide into water, and this is something that distinguishes them from streptococci that we're going to go over in the future, because streptococci are unable to produce catalase enzymes, and so they're unable to break down hydrogen peroxide into water. And an advantage of being able to produce these catalase enzymes, cells in your immune system, like macrophages, produce hydrogen peroxide to kill pathogens, but because staphylococci pathogens are able to produce these catalase enzymes that break down the harmful hydrogen peroxide into harmless water, it gives them resistance to this method of killing them by the immune system. So just one of the ways that staphylococcus are able to fight our immune system. The two main kinds of staphylococci for humans are Staphylococcus aureus, shown here is the kind of golden one. Aureus, I believe, is Greek for gold, and Staphylococcus epidermitis, which is shown here, and it's just a common part of our skin microbiota. So you find it's kind of covering our skin, but it can only really infect us if the skin barrier is broken, like we talked about before with opportunistic infections. Staphylococcus aureus is capable of evading phagocytosis by immune cells as they're coated in a substance called protein A, which binds to the Fc regions of immunoglobulin G molecules, preventing them from activating a complement cascade and opsonizing the Staph aureus. If you're confused about that, I have videos from the medical immunology module that talks about immunoglobulin structure, where we go over the Fc region, which is kind of just the bottom part of the heavy chains. They also have coagulase bound to their surface, which forms fibrin clots that hide the bacteria from the immune system. Plus, the slime/capsule inhibits chemotaxis and endocytosis from leukocytes. Staphylococcus epidermitis, on the other hand, relies almost exclusively on slime to evade detection by the immune system, and after commencement of an opportunistic infection, a biofilm will play a role in inhibiting immune cell function, providing suitable conditions for bacterial proliferation. So we're going to continue Staphylococcus in the next video.